Hello, this is Mr. Merrier with Noteworthy Science. Today we're going to be doing subtopic B3.2 Transport, and I'll be teaching the standard level content. In the curriculum matrix, this falls under B, theme, form and function, and level of organization 3, organisms. Now the guiding questions for B3.2 are somewhat similar to those of B3.1 in that we are exploring a concept at both the animal and the plant level. In this case, what adaptations facilitate transport of fluids in both animals and flowering plants? And then once we know those adaptations, the second guiding question is somewhat intuitive because we're going to be looking at what are the differences and similarities between how this fluid is transported in both animals and plants. At standard level, there are 10 learning outcomes that we'll be going over today. Let's get started. So this first part is going to be a little bit about what is the parts of the circulatory system, and we're going to be looking at adaptations of each one. So I think a good place to start is just to do a very brief overview of the circulatory system. Now again, it's not expected, especially at the SL level, for you to be able to label all of this. I just want to give you a sense as to how this circulation works. So for mammals, we have a 4-chambered heart. 1, 2, 3, 4. And what happens is on one side of our heart, it's deoxygenated blood, which in this subtopic will represent as blue, and oxygenated blood on the left side as red. This is probably a good time for me to explain that in reality, your deoxygenated blood is not actually blue. All of the blood in your body is in fact red. The main difference is that oxygenated blood is a very bright red versus deoxygenated blood is a very, very deep red, near almost purplish. The reason that it looks blue actually has to do with your skin and the interaction that it has with light absorption. So just to be clear, your blood is red, but I'll be representing, and most textbooks will represent blue as deoxygenated blood, meaning blood that has very low amounts of oxygen, and red as oxygenated blood or blood that has a lot of oxygen. As an aside, what's actually quite interesting is that there are actual animals with blue blood. So an example would be horseshoe crabs, and the reason that their blood is actually a very bright blue is because their hemoglobin actually uses copper rather than iron. So I just want to give you a little bit of an overview here. So if I were to follow the path of blood, let's say, here's what we get. Say your lower body here, your blood is now full of CO2. That travels to the heart. The heart then pumps this to your lungs, picks up oxygen, goes back to your heart, and then pumps it all the way back this way. So if you notice, there's actually 2 circuits. So in this case, there's the pulmonary circuit, which is the flow of blood from the heart to the lungs, back to the heart again. And the other circuit, which is the systemic circuit, or the system, or the body system, which is the heart to the rest of the tissues and back. Now, a little bit of a misconception, of course, both of these processes are happening simultaneously. But the way that this is actually one path, 2 paths. In HL, we'll be looking at this significantly more in detail and how this differs in an animal like, let's say, like a fish, where it's not a 2-circular path like this. What are the components of the circulatory system? And it's pretty easy. There's 3 components. There's the heart, the tubes or the vessels, and the blood. The heart acts as a pump. That's physically what's going to push all the fluid, the blood, through your whole body. The vessels act as passageways, right? As like tubes to actually carry this. Today we're going to be looking at those 3 different kinds of vessels: arteries, veins, and capillaries. And then lastly is the actual fluid component, which is the blood. So something that we've already looked at way back in the very first subtopic, A1.1, is we talked about that blood as a fluid is mainly composed of water. So the fluid portion of blood is called plasma. And because it's mainly composed of water, it acts as a really good medium to transport things. So things like ions will readily dissolve. Color substances like carbohydrates or urea will freely dissolve. Zwitter ions, things like amino acids that have both positive and negative charges, will dissolve. And things that can't dissolve are carried by things like lipoproteins that can carry lipids. Oxygen and CO2 are going to be carried in the red blood cells. And of course, you have your white blood cells and platelets, things that are going to be all about clotting. Another big category of molecules that we'll look at later in this course is hormones. Hormones are also transported by the blood. That's how they get from one place to the other place. So having all of this in mind, the next part here is let's go over the anatomy and the adaptations of the different blood vessels of the circulatory system. So the first vessel that I want to talk about are arteries. I think a good place to start with arteries is actually to first dispel a very common misconception. A lot of people have the impression that arteries are defined by what they carry. i.e., if you ask someone, generally speaking, I'll ask them, what's the difference between an artery and a vein? And they'll usually say, arteries carry oxygenated blood, veins carry deoxygenated blood. That is not always true. There are exceptions to this. Arteries and veins are not defined by what they carry. They're defined by their relative direction of flow. So arteries, all arteries, move blood away from the heart. A way of thinking about this is artery away. It's how I always remembered it. So arteries move blood away from the heart always, regardless if it's oxygenated or deoxygenated. It is mainly oxygenated, but there are exceptions. For instance, can anyone think right now of an artery that would be carrying deoxygenated blood? Pulmonary means what? To the lung, right? That makes sense. I'm pushing a bunch of high carbon dioxide, low oxygen blood to the lungs in order to become oxygenated. So the reverse is also true. That's where you're going to get a vein that's actually oxygenated going back to the heart, a pulmonary vein. So these are two exceptions to that rule, and that's why I urge you not to remember arteries and veins by the blood they carry, but by the direction of flow. Now the other major defining thing about arteries, one, they all move blood away from the heart, but their other major defining feature is they carry blood at extremely high pressure. This is probably where a lot of people may be familiar with because of things like slasher movies or horror movies where someone gets cut, let's say in the neck, and blood spurts across the room. That seems like an exaggeration, but that's real. Your blood in your arteries is carried under very high pressure. If we know that blood is carried at really high pressure, now we're going to look at, well, what does the artery actually look like? So I'm going to take a tube and I'm going to do a cross section of that tube. So I'm going to look down the tube. On the left is a micrograph of an artery, and on the right is a simplified drawing that I've done. Okay? So notice that an artery has three major layers of tissue. These layers are named as tunica, which comes from the word tunic, which is like an outer layer of clothing. So it's almost like, it's like I'm layering up for winter. I have three tunics. So if I look from inside out, I have the tunica interna, the innermost layer. That layer is made up of very smooth endothelial cells. The middle layer, notice on arteries, is very, very thick. Now the name here is also fairly intuitive. The tunica media, media for middle. This is very thick layer of smooth muscles, and it also contains collagen and elastic fibers. And the final outermost layer is called the tunica externa. And this outer coat here is made up of collagen and elastin. So now that we know what this is composed of, let's now go into the adaptations. Why does this contribute to its high pressure? What adaptations does an artery have for its function? So all of these adaptations that we're going to talk about all have to do with delivering blood away from the heart at high pressure. So the first one is you'll notice that the inside of the vessel has a very small lumen. The lumen is a term that we'll use for the inside of a structure. So you'll be seeing this term a lot in physiology or anatomy, where lumen refers to the inside of something. So if you look at an artery, the lumen is actually quite small. Now I think a lot of you kind of intuitively know that if you alter the size of the lumen, you alter pressure. Right? The reason I'm pretty sure a lot of you are familiar with this is that if you've ever had like a hose of water and I want to get someone far away, I just pinch the end of the tube and it starts to now flow really quickly. Well, how did you do that? You've decreased the lumen of that vessel, causing much higher pressure to happen, right? So the narrow lumen here contributes to its really high pressure. However, if you're going to move a fluid at very high pressure, you need to support it to make sure it doesn't burst open. So for that reason, the walls of arteries are really, really thick. And I say walls like metaphorically here. They're not really cell walls, right? But the tissue layers are very, very thick. And that helps withstand the really high pressures going on in arteries. The next adaptation that it has is that, well, I said that this tunica media is made up of smooth muscles. Well, what do I mean by that? Well, we've already looked at in B3.1 skeletal muscles. These are muscles connected to the skeleton or to the bone. Smooth muscles, there's obviously no bone here. They are not connected to bone. In fact, these muscles are entirely involuntary, meaning that you cannot control them. They are done through your autonomic nervous system, something that we'll talk about in another topic. So, other example, can anyone think about anywhere else in the body other than your blood vessels that have smooth muscles? Your esophagus, behind your trachea, that's where you're going to move a bolus from your mouth to your stomach, and that's actually caused by contraction and relaxation of muscles all the way down. We'll talk about that in another subtopic. Your intestines, and that's how we can move food all in that crazy way through your intestines. And your stomach. Your stomach is also surrounded by smooth muscle that allow it to churn and allow it to move. So anyways, this is to say that you have muscles. Now they do operate in the same way as any other muscles, which is they work in antagonistic pairs. And when they contract, they shorten. When they relax, they lengthen. So what happens here is that because the tunica media has a lot of smooth muscle, what this means is that your blood vessels can actually alter blood flow and blood pressure. If the body wants to decrease blood flow and increase blood pressure, they're going to do what's called vaso, for vessel, constriction, meaning that your muscles are going to contract, causing the lumen to become even smaller. That will actually reduce blood flow to an area. We'll actually talk about this in the homeostasis subtopic, where when you're really, really cold, your body is going to start to divert blood away from your extremities, like your hands and feet, and move it to the core. Now in the reverse direction, if your body wants to increase blood flow to an area, then they're going to dilate, meaning to get larger. An example of this is that if you've exercised heavily for a little bit, you're going to find that your face is probably quite red. A reason for this is that your arteries have actually vasodilated, because they're trying to actually remove heat more. So they are coming more and more and more away, and it looks like your face is more red as a result. This is actually quite a novel adaptation because it means that blood flow and blood pressure is not static. Your body has control over that. And the last adaptation that arteries have is they have this elastic tissue in the walls. Now, why does that matter? Well, we've discussed this briefly. If you guys remember an adaptation from alveoli, they're surrounded by elastic fibers. Now, what happened is that we said that when you stretch an elastic, you're increasing elastic potential energy. Therefore, when that elastic recoils, it's releasing that energy, right? Well, we can do the same thing with your blood vessels. As pressure is really high in the arteries, it stretches the artery, causing tension, causing an increase in elastic potential energy, and then that recoil will push it through. So it's almost like a rebound pushing blood through this way. So the elasticity of your arteries is also really, really important at not just providing a passive push, but also it resists that high pressure throughout. If you lose elasticity, that becomes more problematic, and that could lead to arteries rupturing and other things like that potentially down the line. Similarly to arteries, veins are not classified by the blood they carry, but by the direction of flow. If arteries were all moving blood away from the heart, then veins are all bringing blood to the heart. So this is how they're defined. Their other major defining characteristic is that blood is being carried at very low pressure. So high pressure away from heart arteries, low pressure towards heart veins. It's almost like a good way to remember adaptations for arteries and veins is if you kind of know one, the other one is effectively the reverse. So actually, when you identify an artery versus a vein, which is a learning outcome, if I were to give you a micrograph like what I see here, it's actually quite easy to tell the difference between an artery and a vein, because arteries will have a very classic circular shape with very thick layers, versus veins are going to have a much larger lumen and a less circular shape because there's not as much things keeping the shape intact. So they have the same three layers that arteries have. They have a tunica interna, media, and externa. But notice the vast difference in the relative sizes of these tissues. The tunica media and externa are much, much smaller than that of arteries. So again, it's quite easy to tell the difference between them. So now let's look at what adaptations do veins have. The first one, again, kind of the opposite as arteries, is they have an extremely large lumen. If you have a very large lumen, that means that blood pressure is low, but blood volume is really high. Like blood flow is very high. So this is why the vast majority of your blood is being carried in your veins and at very low pressure, is this very, very, very large lumen. Now, carrying blood at low pressure causes a bit of a problem. Let's look at our legs, for instance. Arteries have pumped blood very quickly all the way to, let's say, my toes, but now they have to travel all the way back up my legs and all the way back to my heart against gravity at low pressure. That's kind of a big ask. How does it do that? It lacked a lot of the pressure that arteries have. So what's going on? They employ an adaptation that's not seen in arteries, and that is they have one-way valves. So in not all of your veins, but in many of them, especially those in your limbs, if you take a look at, let's say, your legs, throughout you actually have valves. And what a valve is, especially a one-way valve, means that it only allows flow in one direction. So when blood passes past this valve, it will shut, preventing blood to go backwards. So this is preventing blood effectively from pooling in certain parts. In fact, You've probably seen what happens to those individuals whose valves become faulty. It's something called varicose veins. If you've ever seen veins on individuals' legs that look like spider-like and all over the place, that's a problem with the valves, causing blood to pool in areas and causing the vessels to become more distended. That's what's going on here. Now, this last part here kind of works in conjunction with the valves. Notice that when we looked at veins, their walls are relatively very, very thin. This is actually advantageous because your veins are surrounded by skeletal muscle. And so what happens is that, let's imagine I have blood right here in between two valves. I can contract these skeletal muscles. By contracting it, it actually opens this valve and pushes blood forward. So believe it or not, but something like your calves and you actually flexing your legs promotes blood flow through your body. So you may also see people, like let's say on long flights, may want to walk around or do something because it's actually a good thing, because the idea of sitting down and not moving for a long time actually can impede blood flow. So your skeletal muscles actually contribute to blood moving through your veins. Now, if your arteries move blood away from the heart and veins bring them back to the heart, when does a vein become an artery and vice versa? When do they change names? Well, the connection point is capillaries. So arteries are connected to veins via capillaries. Arteries and veins are only all about transport. You can consider them like highways. So let's look at a layout of a very big city. Where traffic is going to be the highest and the fastest is going to be on highways. So these would be like your arteries and your veins. If I want to go from a highway to my driveway, let's say, I can't go from a highway directly to my front door. I have to go to smaller and smaller and smaller roads. And the same thing is true for your blood vessels. Your arteries will branch off into smaller arteries called arterioles. We've seen this suffix before when we looked at gas exchange, where I said bronchus, bronchiole, which is a smaller version of that tube. So I have artery, arteriole, and then this arteriole then starts to form massive amounts of branches, and this is your capillary. Sometimes referred to a capillary bed if it's very, very branched here. This zone is where exchange happens. All that stuff that we just said here, how is this going in and out of cells? All of that transport is happening here, where the nutrients like oxygen, glucose, amino acids, whatever, that's coming out here. And then all the waste products from your cells, things like carbon dioxide, urea, is entering your capillaries. After they leave the capillaries, we can now call them veins, specifically very small veins called venules, and then these become very large vessels called veins. So capillaries are the connection between both of these vessels. Now, unlike arteries and veins, their structure is very, very simple, and it's actually quite different. Take a look at this micrograph. This should show you just how small they are. This is a single red blood cell. A capillary is only marginally larger than a red blood cell. In fact, they're so small that red blood cells have to pass through in single file. So they're incredibly tiny. If they're this small, that means that the blood pressure must be ridiculously small, as well as the blood flow. We want to look at their structure and then explain how their structure is adapted to their function. So let's look at their structure first. Notice we've lost a lot of the layers. There's no tunica media. There's no tunica externa. In fact, there's only a single layer of cells called endothelium. And outside the endothelium is a another barrier called the basement membrane. That's it. That's a capillary. And what you'll also notice in many capillaries, this endothelium is not even contiguous. It actually has gaps or pores. And we call these fenestrations, which comes from, I believe, the Latin, or at least I know it in Italian, which means window. Finestra. So fenestration is like little windows, little openings. So let's look at how are capillaries adapted to their function. What are the function of capillaries? Exchange. So if we think back to B3.1, when we were talking about how do I promote exchange, well, I want really, really tiny distance that things have to travel. Well, there's extremely small distances. Look at the distance from red blood cell to cell in your body. It only has to cross a handful of membranes and it's there. The distance is incredibly small, especially because the red blood cell is only slightly smaller than the diameter of a capillary, which means it's right there. So the distance of exchange is really, really minimized, which highly accelerates the rate. Another one is, as we've discussed also in B3.1, it has an extremely high surface area, and that's due to the multiple branches that it makes. Exchange rate is extremely, extremely high because there's so many different paths for gas exchange and material exchange to occur due to the very, very, very high surface area. And then finally, these pores or fenestrations allow tissue fluid and materials to very easily exchange. I mean, what's faster than going through a few membranes? Going through 0 membranes. So we can just pass directly through these pores. Now, it should be noted not every single one of your capillaries has holes. They are in key areas, but many of them do. So overall, what you should get out of this is, do you understand the main components of the circulatory system? Could you tell me the anatomical features of arteries, veins, and capillaries? And could you tell me how each of those features are adapted to their function? So the next thing is going to be about certain measures that you're almost certainly already familiar with. And the first measure that we're going to be discussing is that of pulse. So what exactly is a pulse? So the word pulse really just means like a regular rhythm. So really it's the regular rhythm throughout your arteries, right? Because as we've already talked about, the flow of blood through the arteries has a pulsile nature, meaning that because of, we talked about the ability of the walls of the artery to stretch and then recoil, that's causing this pulsile nature. Because you can feel the actual bulge of your arteries, that's really what a pulse is. With every heartbeat, your arteries are in fact stretching slightly, and that's what you're measuring. So we're actually going to start today with just understanding how we can actually measure a pulse, both manually and with a device. So the easiest way to measure a pulse is your carotid pulse, and that is your carotid artery right here. Now, I have a little bit of experience about this. Back in the day, I used to teach first aid and I used to teach swimming, so being able to do pulse checks is quite necessary, but I would say that this is a skill that everybody should be able to do. A big mistake that students or just people make when they're taking a pulse is they'll try to take it from the side over here, and that's going to be very difficult. You're not going to feel anything over here. The easiest way is to find your trachea right here, and you're going to go into that little gap next to your trachea, and you're going to feel a very clear pulse this way. Now, the way that you can determine a pulse rate would be how many pulses over a given amount of time. This is usually expressed in per minute, but this doesn't actually mean that you have to take the pulse for an entire minute, right? You can take it, let's say, over 10 seconds and then multiply it by 6. So let's all try that right now. Find your pulse and then use a watch or whatever, and then take it over 10 seconds. Another way to check a pulse, other than your carotid pulse, is your radial pulse, which means your wrist. So in this case, again, you're going to use 2 fingers and you're going to try to feel it right over your artery. So if you look at your wrist right here, you're going to take it right here and you should feel, again, a pulsile motion right in the wrist. Pulse rate would be the frequency of pulses over time. In effect, this is being used as an indirect way of measuring heartbeat, because I'm not directly measuring the heart, but by measuring the expansion effectively of your arteries, it effectively means the same thing as your heartbeat. So the pulse rate and your heartbeat are virtually the same because they come from the same thing. Now, if we wanted to measure this using a digital apparatus, then something that we can use is right here. I borrowed this from the school nurse. And this is what's called a pulse oximeter. So I just kind of want to explain to you how a pulse oximeter works, and then we can practice using it. So I just want to go into a little bit to the theory as to how this works. So what you do is you take a pulse oximeter like a clip, and then you clip it over a finger. And then you would just turn it on this way. And then what happens is that using the pulse oximeter, ooh, fancy. So what you'll see is you'll see not only a pulse rate, but you're going to see a pulse out graph. Now, how exactly does this work? Well, the way that this works is that it's actually quite cool, because if you do a cross section right here, and an individual's finger is right in here, here's the top part of the clip, here's the bottom part of the clip, it's actually shining light. And specifically, that light is shining through the skin, into the artery, into the vein, and then being detected back out here. It's almost like a spectrometer in that what it's measuring is absorbance, right? So as light is shining through, it's taking an absorbance. Now, how the heck does this tell you what percent of oxygen is there? How does this work? Well, oxyhemoglobin actually has a different absorbance than non-oxyhemoglobin. So what we can do is you can actually measure how much of that is being absorbed, and that can be used as a proxy for how much oxygen saturation does your blood have. A pulse oximeter not only tells you your pulse in real time, but it also gives you an estimate as to the oxygen saturation. What it's going to produce is a graph that kind of looks like this, and actually this will produce it on the screen as well. It's something called a plethysmograph, or pleth for short, and really what this is the absorption over time, and that effectively translates to a pulse rate. And so what's going on here is you can see how is that absorbance changing over time with respect to pulsatile blood in the arteries. It also should be noted that what exactly is controlling your pulse rate? Well, for good reason, you can't directly control your pulse rate. That's good. I don't, to be honest, it's control that I don't want. I don't want to control my heartbeat. I want the heartbeat to happen on its own. You can indirectly control your heartbeat by, for instance, breathing really fast or doing really high physical activity, because really what you're telling your body is, I need a lot more oxygen, and then your body is going to respond in turn by increasing your pulse rate, or your heart rate rather. So generally speaking, your sympathetic nervous system is what's going to cause your heart rate to increase more, and your parasympathetic system is what it's going to cause it to slow down. We're going to be looking at both of these methods of control in integration of body systems later in this course. Okay, the next portion that we're going to go into here is, well, it's slightly sad, and that is heart disease. So cardiovascular heart disease, or coronary heart disease, in fact, is the leading cause of death worldwide. So what exactly is it? And I think to really understand what this is, we have to first do a little bit of anatomy. If I'm talking about coronary heart disease, what does coronary mean? So coronary refers to the heart, right? So if I'm a coronary surgeon, I'm working on a heart. So far, everything that we've talked about with arteries and veins, we've said arteries move blood away from the heart, veins bring it towards the heart. But something that you may have not considered, well, what's giving the heart blood? The heart is a muscle that is working forever until you're dead. It is in need of a tremendous amount of oxygen and gas exchange. So what is actually feeding it? So there's actual arteries and veins on the surface of the heart itself. So as blood is coming out of the largest artery here, the aorta, it's coming from here and feeding the rest of the heart muscle oxygen and causing gas exchange to occur in those tissues. When we're talking about coronary heart disease or coronary problems, we're dealing specifically with those on the heart. So that's what we're discussing here. A major issue that can occur is something called atherosclerosis. And that's a fancy way of saying it's the hardening and narrowing of arteries. Now let's think back to what the adaptations of arteries were. We said that arteries, we would want it to be very flexible in order to accommodate high pressure changes and to allow for recoil. Now look at this disease, hardening and narrowing of arteries. If I narrow arteries even more, what will that result in? Pressure will go even higher than normal. And secondly, imagine now I have more pressure from narrowing arteries, but also they're hardened. Why might this be a problem? It's not as flexible. So now I'm in a situation where the tube is at even higher pressure than normal, and I've lost or at least reduced the capacity to resist that pressure. This is a recipe for disaster here. This is a big, big problem. Atherosclerosis, what does this actually mean? So this comes from atheroma, which is a fancy term for plaque. Plaque, you've likely heard from a dentist point of view, where you may have plaque buildup on your teeth. It really just means a deposit. So what's going on here is you may get a deposit or plaque buildup within the arteries themselves, and that is causing an increase in pressure. So this atherosclerosis can lead to coronary heart disease. And specifically what that means is that the coronary arteries are narrowing, and this may lead to an occlusion. An occlusion means a complete blockage of that tube. So before we go into that process, let's go over risk factors. So anytime that you're dealing with some kind of disease, it would be inaccurate for me to say, if you do factor X, you will get this disease. That doesn't happen. But what could happen is we can say what factors increase the likelihood of developing this disease, right? So we're going to be going over the key risk factors associated with coronary heart disease. The first one, and very, very prevalent one, would be smoking. Smoking is strongly correlated to coronary heart disease. Now, smoking, for a number of reasons, is going to cause problems. In addition to the damage that it can cause to your arteries, it can also lead to hypertension. What does this mean? This means that your blood pressure is chronically high. Chronic means over a long period of time. The opposite to chronic would be like acute. So chronic high blood pressure, that's a problem because that means your blood vessels are consistently at high blood pressure. Another risk factor here is hyperlipidemia. Hyper, again, meaning high. Lipid, meaning that there's a high amount of lipid being brought up here. This is a result of high LDL. What LDL stands for is low density lipoproteins. And so in your body, you have both LDL and HDL. They effectively do opposite roles. LDL cholesterol delivers cholesterol from your liver to the tissues, whereas HDL takes excess amounts and brings it back to the liver. So one is kind of like a delivery from the place where it's stored in the liver to where it's needed, and the other one is picking up any excess. So to say outright that LDL is bad is not true. However, if you were to consider both LDL and HDL as a balance, you would want more HDL than LDL, and that effectively will prevent a buildup of these lipids within your actual blood vessels. If you have a lot more LDL than HDL, that means there's more things delivering than taking back. That's what's going to lead to an increase. So, saturated fatty acids that are derived from animal sources, these actually promote LDL more than HDL, and the one that has the highest impact is trans fats. Trans fatty acids promote significantly LDL. This is why they're sometimes referred to as bad fats. Unsaturated fatty acids that are derived mainly from plant sources, things like plant oils, these actually promote HDL. So these are the good fat. The reason why dietitians would encourage more unsaturated fats versus saturated fats is because unsaturated fats promote HDL over LDL. If your diet consists largely of saturated and trans fats, then you are more at risk of this because your body is effectively promoting more LDL than HDL. Now, hyperglycemia, this is something that we'll discuss later when we do homeostasis, but hyper, again, means elevated, and glyce here means sugar. So hyperglycemia is when your sugar levels are significantly elevated. The most common reason for this would be someone who is diabetic. This also is a risk factor for coronary heart disease. And a very large risk factor is obesity. Obesity has a very strong correlation with coronary heart disease, and obesity can be due to a lot of genetic factors, but it could also be due to lifestyle changes, things like a sedentary lifestyle, meaning very low physical activity, and it could also be strongly related to your diet. So, taking all of these risk factors, there's another unfortunate risk factor here, and that is just being male puts you at higher risk than being female. Males are more predisposed to coronary heart disease. It impacts males more than females. And your risk factor increases with age. So the older that you are, you become more and more prone potentially to this happening. So here's a step-by-step process of the development of coronary heart disease. I tried to draw it from left to right. So firstly, and just for clarification, what exactly are you looking at here? This right here would be the lumen of your blood vessel. This would be the actual tissue layers beneath. So this would be the tunica interna and then the tunica media. Underneath is basically an overall cross-section of what's happening to your blood vessel over time. So at the beginning, if you have any of those risk factors, what could be happening is that they are causing damage to actually your endothelial layer. So they're physically damaging the cells here. If that's happening, then that means that LDL, or low density lipoproteins, can actually enter into the tunica intima. Once this happens, your white blood vessels are like, whoa, 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 what's going on here? They should not be here. So your white blood vessels, specifically your phagocytes, something that we'll talk about when we go into the immune system, they're actually going to start to engulf and try to destroy this LDL and process this LDL cholesterol. Once that happens, they're going to build so much that they'll literally they'll literally consume as much until they burst, restarting the cycle again. So you get this positive feedback loop where they're trying to take as much, they take so much that they burst open, suddenly there's a bunch more there that will promote even more white blood cells to come to the area. And this whole thing is what's going to cause a buildup of this plaque. That's what this plaque is. Now, a big misconception that students, and I'd say the general public has, is I think a lot of people assume that this plaque buildup is happening in the lumen. It is not. And I showed that here. Notice that the plaque buildup is actually happening within the tunica intima, not the lumen yet. So this is all still below, but as you notice, with the more and more and more plaque buildup, you're starting to get a bulge. You're starting to bulge in, and then what's that doing to the lumen? The lumen is starting to get smaller and smaller and smaller because of this buildup of plaque within. So this is going to continue to grow. Now, to try to mitigate any breaking of this, your body is going to start to create what's called a fibrous cap. So this is a layer that is made up of fibrous proteins to try to resist any of that breaking right there. So hence, this is where the hardening of your arteries come from, is that you have this fibrous material now that's trying to block this from occurring. But at a certain point, what could happen, and this is where it becomes really catastrophic, is if this plaque actually bursts open. If this becomes so large that it bursts open, well, your body is going to treat this like a blood clot. So this is another process that we're going to be looking into when we go into the immune system. But the too long didn't read version here is that this is the same process that would happen as if I cut my skin. Your body is going to start to induce a number of changes to seal that gap. And they're going to try to do the same thing here. But remember, I'm already in a narrowed environment. If this breaks open, they're now going to cause a clot here. You now have a complete occlusion. I now can't deliver oxygen anymore. So, or anything really, the entire blood flow has been cut. So this is how an occlusion can happen. And hopefully you can understand, why did the lumen narrow? Why did the artery actually become hardened? And how can this lead to an occlusion? So who cares? What are the consequences of this? Is this really that problematic? Yes, this is very problematic. So the idea is that I went through this process fairly quickly. This would not happen over the course of a day. This would happen over the course of years, potentially decades, right? So this is a slow, ongoing process. Now, the idea, and this would be what would be considered a progressive disorder, meaning that it's worsening over time, potentially very slowly. Now, what are some very serious consequences that can happen as a result? Well, one, before there's a full occlusion, even at this stage, with a narrowed lumen, this would increase blood pressure. This is what would lead to hypertension, i.e., this is a major risk factor for this happening in the first place. So you can see how the two are feeding back into each other. And this is actually also quite common in older men that have high blood pressure, and they may have to start to take medication to lower their blood pressure to try to mitigate this risk factor. Secondly, as this is being repaired with this fibrous tissue, it's hardening your arteries. Again, this makes them more prone to potential bursting. They have less ability to stretch and recoil. This is, again, a fairly big problem. This makes them more prone to damage. Thirdly, if you actually get that plaque rupturing, then the idea is that that clot, called a thrombus, actually impedes blood flow. This should be quite clear why this is problematic. The oxygen and nutrient delivery to tissues now is going to be completely impaired. And remember, if this is all happening in your heart, this is a big problem. This means your heart muscles are no longer getting any exchange. If this happens in the heart, then your cardiac tissue is literally going to start to die. Your heart tissue is literally dying because it's being starved of oxygen. And this is what a heart attack is, or the proper name for a heart attack is a myocardial infarction. This is why it's imperative to get medical help for someone undergoing a heart attack immediately, because the longer you wait, the more heart muscles are dying. And heart muscle are not tissue that your body can repair. Once heart muscle death occurs, it remains dead. So the sooner we get help, the better. Lastly, this may not only affect the heart. If this clot is formed, it could break off and travel elsewhere. Once that clot breaks off, it's now called an embolus, by the way. But if that's now getting pumped and transported elsewhere in the body, it can actually cause an occlusion elsewhere. And this is how strokes can happen. Or this is now causing an occlusion in my brain. This is now starving brain tissue of oxygen. That leads to a stroke. So it's not just necessarily the heart that can get impacted. It could be other tissues as well. Now, I've mentioned earlier the idea that those risk factors are correlated to coronary heart disease. What do I mean by correlation, right? So when two things are correlated, or two variables are correlated with each other, it's effectively a degree of association between two variables. So what we try to do a lot is we try to assess if a risk factor is correlated to a particular disease, meaning the more and more this risk factor is prevalent, the higher the incidence of this disease. You can then claim that these two variables are very strongly correlated. A lot of epidemiological studies, we've been able to correlate all those risk factors with causes and trends of this disease, meaning that when there's more smoking, when there's higher levels of obesity, when there's more hypertension, when there's hyperglycemia, all of these had a higher incidence of coronary heart disease, thus they are correlated. Now, the way that we can statistically assess correlation is something called the correlation coefficient, or R, or Pearson's R. So what this is it's quantifying both the correlation, the strength, and the direction, so to speak, of a correlation. So a correlation can be positive or negative. Now, the R value actually tells you all of that. So if you had a R value between -1 and -0.5, that would be considered a very strong negative correlation. If you had a R value between -0.5 and -3, that would be considered a moderate, and a very weak correlation would be between -0.3 and 0. Now, the opposite is also true, or the converse is also true for positive. So it's almost the closer you are to 1 being a perfect negative or positive correlation, anywhere closer to 0 becomes weaker and weaker and weaker of a correlation. I can't possibly talk about correlation without talking about how correlation can be misused and misunderstood. So there's a very famous saying in statistics, which is correlation does not equal causation. And that's a very, very important factor to consider. In fact, there are so many different examples as to I can just look for 2 random variables and find really nice correlations between the two. Ridiculous examples of them. Just because 2 things are correlated does not mean that they're causational. Or it could be either due to a third factor. You could be looking at this in the other way around. There's a lot of ways of misunderstanding correlation. Now, this is not to say that I'm saying all of these things are correlated, they're not causational. They are causational, but just doing a correlation is not enough to do that. You would then have to do another study to determine, is this actually causing the other thing? So at first, we can do large-scale studies, look at a country's, let's say, incidence of coronary heart disease, and then assess factors like obesity, age, sex, like all kinds of other factors here, or level of smoking. And then we can really see, okay, this factor and this factor are clearly correlated. Now let's do a study to realize, or to try to deduce, why is this factor potentially causing this? So you'd have to take it, correlation is, for me, is almost like the first step. These 2 things may be very related to each other. Let's figure out why these things are correlated to each other. So that's a really important factor to consider here. Similarly to B3.1, where we looked at a process, in that case gas exchange, at both the animal and the plant level, B3.2 Transport is going to be looking at, okay, how does transport happen in animals? We just did that. And now we're going to be looking at it, how does it happen in plants? So when we say that fluids or things are being transferred in plants, obviously they don't have blood or blood plasma. It's mainly carried as water in one of 2 vessels, that being the xylem and the phloem. So a good place to start with this subtopic is the anatomy, right? So similarly to when we looked at the cross-section of a leaf, when we did a dicot plant, again, we're sticking with dicots here. So we're going to be doing a cross-section of a stem of a dicot plant. Let's take a look at the various tissues that are present. Similarly to the leaf, the outermost layer we said was called the epidermis. Epi for outermost, and derma, again, technically means skin, but in this case it's the outermost layer. So in this case, for a stem, the epidermis is really going to be the source of protection for that plant. So it's going to provide protection, and it has also a waxy cuticle, which is composed of wax or lipids, to prevent any water loss coming from here. Just below is going to be the cortex. And this is another word that's also used not just in plants, but in animals as well, such as the cortex in the brain or the cortex in the kidney. Cortex typically means the outer layer. So like the cortex of the brain versus the medulla, which is more inside. So the cortex here would be cells that are used for support, photosynthesis, metabolic processes, nutrient storage. It's basically a lot of stuff is happening in the cortex here. And that's why this tissue layer is actually quite big. Then we have a vascular bundle. So what do I mean by that? Well, if you really zoom in here, this is the vascular bundle. And vascular or vessel means essentially those tubes. And in this case, we have xylem and we have phloem. Now, similarly to when we looked at a cross-section of a leaf, the xylem was placed above the phloem, if you remember, in that vascular bundle. Well, the same thing is true here. The xylem is facing the interior versus the phloem is facing the exterior. This, I'd say, is a fairly common source of confusion because you may be asked to label a cross-section of a transverse stem. So you should be aware of the relative positions of each. Can somebody explain to me why I don't just have one large vessel here for xylem and one large vessel here for phloem? Why do I have a bunch of small tubes all very close to each other? We've looked at this in A1.1, actually, when we looked at cohesion, adhesion, capillary action. The smaller and smaller and smaller the tube becomes, the greater the surface area to volume ratio of inside that tube, right? And the two major factors at play here are the adhesive forces, or the attraction forces between the water inside the tube and the wall of the vessel, and the cohesive forces and the weight of that water itself. So if the adhesive forces are greater than the cohesive forces, you're going to get this upwards pull of capillary action. But you can only really do that when the tube is really small. So this is why you have a bunch of these little tubes all connected as opposed to one big xylem and one big phloem. So the xylem and the phloem, again, have two different roles. The xylem is all about transporting water, and whatever would be dissolved in that water would be mainly ions. Now the phloem also will have water, but it transports mainly sugars and nutrients from metabolism. We would colloquially call the contents of phloem sap. So that this sugary, watery substance, that would be within the phloem itself. Now, similarly to when we looked at the cross-section of a leaf, you are expected to be able to do a plan diagram. So let me show you how to draw a plan diagram of a stem. So let's do a circle. Now the uttermost layer, let's do a circle inside the circle. And then you're going to do a series of little ovals all within the inside. Then an easy way of separating them is you can just do a line all the way in the middle. This would be the epidermis, so that's on the outermost section right here. Here you would have the cortex right there. Then right here would be the vascular bundle. Then I would independently label both the xylem and the phloem. So this part specifically is the xylem. This part is the phloem, just like that. And actually, that would be sufficient for this diagram. Now I have a few extra labels in my notes here, and that being the pith, which is the inside part. You don't need to be familiar with that, but if you've ever seen the cross-section of a plant, it's the really hard part. It's a lot for structure. You don't necessarily need to know that in this course, but that's what this innermost part is. It's called the pith. And actually the partition between the xylem and the phloem is something called the cambium. Again, that's what this line across is, but you also don't need to draw that either. This first part was all about the distribution of tissues in a transverse section of a stem, just like this. Now, It's one thing to know anatomy, but it's a whole other thing to know of how are these structures adapted to their function. So what adaptations do xylem have in transporting water? The first adaptation here is actually quite odd, and that is xylem are kind of in a weird category in the sense that they're not alive, actually. And you can kind of tell that because they don't have anything. They don't have a cytoplasm, they don't have a nucleus, they don't have any cell contents. They're really just glorified straws, really, is what they are. So the idea here is that they don't have any of these contents. Now, why is that adaptive? Well, they're a straw, right? I don't want a bunch of stuff in a straw if I'm trying to take from one place to the next. Being completely hollow without a jelly-like structure in the middle with a bunch of organelles, none of that's needed. The whole role of a xylem is just to provide a tunnel for the passage of water. So by lacking all of those things that normally a cell has, that in and of itself is an adaptation. It's actually quite useful. So this allows this really rapid, unimpeded flow of water up through a xylem. Another adaptation is if you look at a cross-section here, is you see that there's actually gaps where water can flow from one xylem to another xylem laterally. And if you look at this from another perspective here, these are what are called pits, or little openings here. Now these pits, what this allows for is that it allows adjacent vessels to be connected. This is quite important because it allows the lateral movement of water. So even if water was at this one point of a plant, it can actually move laterally to another part of a plant through these pits. So it allows a more uniform flow of water through a plant. The final adaptation that I want to talk about here is that xylem are not just composed of classic cellulose, they also have an additional structure for strength, and that is called lignin. So lignin, as a polymer, the cell, it basically, in a lot of, not always, but in a lot of xylem, they actually will brace the inside of a tube in this helical structure, as I've drawn here, and they're almost acting like a brace. Now, you might be like, well, is this brace for high pressure? No, it's actually preventing collapse from low pressure. Consider transpiration, right? We have very low pressure in the leaves, very high pressure in the roots. I am confident that I can get an everyday example to help explain this to you. The difference between a really bad straw and a good straw. If you're sucking really hard on a straw, and that straw will what? It can collapse, and suddenly it doesn't work anymore. That's what would happen with the enormous pressure differentials of a plant. Whereas you have huge pressure differentials, and that would actually, at low pressure, would actually cause the xylem to collapse in and on itself. We don't want that. So lignin is actually used to bolster the sides to prevent that from actually collapsing. So I'd say this is a fairly prominent misconception, because I think a lot of times when we think about structural supports, we're thinking that it's blocking against bursting from high pressure. But in this case, it's preventing low pressure, causing it to collapse. The other thing that's actually quite important for it is that lignin as a molecule is hydrophobic. And so the idea is that it prevents the xylem from absorbing water. We only want it as a passageway. Again, you've likely seen the difference between a paper straw and a metal straw. If you keep using that paper straw over time, the straw itself is going to absorb that liquid. That's a problem. We don't want it. So in this case, I don't want the xylem to absorb water itself. I just want it basically as a tunnel. So this lignin is also acting as almost like a coat to only allow water through and not it being absorbed. We've alluded to this process in so many other subtopics in this course. Let's finally talk about it officially, and that is transpiration. So transpiration really is just the passive flow of water from roots all the way to shoots, right? So it's going to enter into the roots, and it's going to exit out of the stomata in leaves. So this is a one-directional flow. Why is this process so crucial in plants, right? Why do it? Firstly, it is a cooling mechanism for plants. As we've discussed previously, water has a very high heat of vaporization. That means when it evaporates, it requires a lot of heat or energy to evaporate. So when it evaporates from a surface, it's in effect pulling energy or heat from that surface. So transpiration is actually a cooling mechanism for vascular plants, making it a really effective tool for this. Secondly, it allows transport, right? How is a plant meant to bring ions that it absorbed from the roots all the way to the leaves? How is it doing this? It needs some medium to move it. And we've discussed back in A1.1 that water is an excellent medium for this process. So this process of transpiration allows things to flow through and be delivered in a plant. And then finally, from an ecology point of view, it's the main driver of like weather around it. All that water that's exiting through transpiration, that ultimately is going to condense into clouds, causing rain. And in fact, you start to see huge shifts in weather after deforestation happens. Once you lose a bunch of plants, you no longer have that transpiration there, and that whole area becomes a lot more dry as a result. Plants are a really big driver of things that you may not even have considered, things like the weather of the surrounding area. So how exactly does this happen? Let's take this step by step. You can either look at this process from roots to leaves or leaves to roots. I'm going to explain it from the leaves perspective, but understand that transpiration is always going to be unidirectional, meaning it's always going to flow from roots to the leaves. It's not going to flow in the reverse direction. But I'm going to be describing it starting with the leaves because I think it's easier for me to do so. So the reason I'm starting with the leaves is that if I have a movement due to a pressure differential, I want to know what the driving force of that pressure differential is. And in this case, the driving force is water evaporation. And that is almost always caused by the sun. So the sun is causing water to evaporate, and we've discussed this in B3.1. So we have water as a liquid evaporating into a vapor. That is then coming out of the stomata, right? The guard cells would be turgid and open. Water is coming out. This is now causing a negative pressure potential or a suction force inside of the leaves. So here is my zone of very, very, very low pressure right here. Then another aspect that we've looked at in A1.1 and throughout this course is capillary action. So water is going to be able to be driven up the xylem, both due to cohesion, causing water to attract itself, but more importantly, adhesion, allowing water to be attracted to the polar xylem on either side of it. So it's now moving up this way. So capillary action is the upwards pull of water up the xylem. And then, well, where is water actually entering the plants in the first place? And that would be the roots. If I want water to enter a tissue, then what do I do to that tissue? So this is actually something that we'll look at more in HL. But what roots are doing is they are effectively pumping themselves full of ions from the soil. That's causing them to be relatively hypertonic to their surrounding soil. Because they are hypertonic to their soil, water is driven from high water potential to low water potential, or from a hypotonic to a hypertonic environment and being driven into the roots. Now take a look at the structure of these root cells. What do you notice about the structure? How might the structure of this root cell right here, called a root hair cell, be adaptive to its function? This long extension here, hence its name hair cell, is a massively increased the surface area to volume ratio of this entire cell. But with more surface area to volume ratio, that means more membrane, more surface for water to enter via osmosis. So they have a very, very high surface area to volume ratio here. This is where water is being driven inside. And actually, water has two different mechanisms of moving throughout the tissues. They can either move through the space in their cell walls, and we call that the apoplast pathway, or it can actually move through cytoplasm, and we call it the symplast pathway. So water has multiple ways of moving from inside into here. So for context, if I were to do a cross section here, this would be like outside of the plant going into the core of the plant, right? That's what we're looking at here. So the last thing that I want to talk about is, now that we understand how transpiration happens, what's going on in the stems, let's do the same thing, but let's look at how our tissue is distributed in the roots. So let's take a look at that. Some similarities between the roots and the stems is that they have a lot of the same tissues, but they're organized in slightly different areas. Now again, it should be noted that what we're looking at here is in the root of a dicot. So when we looked at stems, roots, and leaves, we are dealing with dicots. So that specifically is what the course is after. It is worth noting that the distribution of these tissues is different in monocots. So we're only dealing with dicots in this course. So again, similarly to stems, the outermost tissue would be the epidermis. This is actually, if I were to zoom into here, if I were to really zoom in, this is where all the root hair cells would be. This is what would be facing out, right? Because remember, I'm doing a cross section of a root. So the outermost part, this is where the root hair cells would be, facing out into the soil. So for context, this would be the soil. This is the cross section of the root, which means water would be moving in this direction from outside to inside. So water is entering into the epidermis, then it's flowing through, again, either through the apoplast or symplast pathway through the cortex. And this is really just bulking out the root for support. It's really just water passing through it. It's not doing that much necessarily. It's, again, metabolism is of course happening here, but it's mainly for structure, support, nutrient storage, etc. And then finally, we're getting to the vascular bundle, because remember, the whole point of this is to bring water from the soil and deliver it to the xylem. The difference is that the vascular bundle is located right in the center. So in this part right here, we have the xylem in this like cross-shaped structure, and then all around the xylem are the phloem right here. So as you can imagine, drawing this is actually not too bad. So let's draw this step by step. Here I have, again, a circle, no big deal. Here's the epidermis. Then we're going to do a circle roughly in the center, and then just connect the sides here like this. Just like that. And that's it. We're done. Outside would be the epidermis. Inside is the cortex. This whole thing together is the vascular bundle. This larger cross shape here, this is the xylem. And then here would be the phloem right here. So again, not too bad. Pretty straightforward. Let's go over the standard level learning outcomes for subtopic B3.2 Transport. The first learning outcome had to do with how our capillaries adapted to their function, i.e. how our capillaries adapted to exchange materials between the blood and the internal and external environment within organisms. We then dived into the structure of both arteries and veins, and specifically, if provided micrographs, students should be expected to distinguish either of these structures. In addition to the structures, the follow-up learning outcome here is really understanding how arteries are adapted to their function and how veins are adapted to their function. We then looked into how pulse rates can be measured, both in traditional mechanisms such as using your fingers, or using digital ones such as oximeters. Finally, with respect to animals, we looked at the causes and consequences of the occlusion of coronary arteries and the idea of coronary heart disease. This then led into the discussion of correlation and causation. And just because variables are correlated does not necessarily mean that they are causational. And understanding this distinguishing point is actually really, really important. So the second part of these learning outcomes really explored how transpiration occurs in plants. And so the major process that we discussed was transpiration, and how water can be moved from the roots all the way to the leaves. We then explored the adaptations that xylem vessels have in order to facilitate this transport during transpiration. And then we explored the distribution of tissues both in the stems and the distribution of tissues in the roots. And for both of these, students should be able to draw a plan diagram showing the distribution of these different tissues. If you wish to learn further, which I always encourage you do, then this last page is a good place to start. These are the textbooks in which I consulted and their relevant page numbers in creating these notes. The 3D models here are actually extremely interesting in actually exploring both the anatomy of humans and the heart, but also in terms of plant transpiration and how these processes are actually occurring in a three-dimensional point of view. And these simulators and interactives are truly invaluable in helping to understand the different anatomical features and how these processes all work together. Now, it should be noted that many of these are more strongly related to the HL learning outcome, such as the cardiac cycle and being able to label the heart, but they are still really valuable resources in really understanding how these processes work. And then finally, these are some pretty interesting articles related to other learning outcomes that we've discussed, such as heart transplantation and what technological improvements are there. And then these really deal into cardiovascular disease and the certain risk factors associated with it. Finally, I would always encourage students to not just learn for the sake of a test, but learn for the sake of learning, learn for enjoyment.